All right, I'm going to dive right in because you guys came to see Allan, not me. Allan Drummond is a polymath, and I don't mean that just in the STEM sense. I mean that in the STEAM sense. He's actually makes some very beautiful science-oriented metal work that has shown on his own exhibits and has actually made some pretty serious bank in some cases. So ask him about that afterwards. Like many of our genetics colloquium speakers, he has a bachelor's degree in mechanical and aerospace engineering, so pretty typical from Princeton University. He then went actually to a Trilogy software company, kind of in the dot-com era of the 90s, and ended up performing several roles there. Ultimately was the director of their university with 180 employees. He then decided he was making too much money and decided to go to graduate school. And fortunately, he ended up at Caltech in the lab of Nobel Prize winning, Nobel Prize winner Francis Arnold, known for, of course, protein engineering. And there is where I first became of his work, which actually won the top thesis award at Caltech. And he really performed seminal work that showed actually that the largest explainable percentage of the rate of protein evolution was actually due to various things related to protein expression levels. And if you're thinking about the problem from a biophysical standpoint of what it takes to express and translate and fold a protein, that makes a lot of sense. But this is not how evolutionary biologists were thinking at the time at all. And it really created a lot of waves, but I think it has held up over time. He then went off to do one of these super postdocs as a Bauer Fellow at Harvard for 5 years. And then, unbeknownst to me until last night in 2010, he and I were competing on the job market at the same time for many of the same jobs. I'll leave it to you to decide who won what, but he ended up at University of Chicago in the Department of Biochemistry and Molecular Biology, as well as the Department of Medicine. He has won many awards, including he's a Sloan Fellow in Computational and Evolutionary Molecular Biology. We overlapped, and that's, I believe, the first time we actually met in person as Pew scholars in the biomedical sciences. And then just to kind of emphasize the polymath nature here, I'm going to read a list of societies that have given him named lectures or awards. And I defy you to find a pattern here. The American Statistical Association, the Biophysical Society, the Marine Biological Society, and the MD Anderson Cancer Center. And he tells me now that he has a project he's been working on for several years that is going to make a splash in theoretical physics, and that's what he's going to tell us about today. Oh, no, stress granules, rethinking.
Holy cow, Chris, come on. So first I have to say, it's a complete joy to be here. I was here a couple of years ago for biochemistry. And so I received this note basically saying, do you want to come to UW-Madison to give a talk? And I was like, well, I sort of feel like I already did. Then I realized Chris was asking. I was like, okay, I'm absolutely saying yes. Chris is one of my favorite scientists in the yeast community in particular because he knows everything and he's willing to share his knowledge about everything. And so when we're doing our studies that I'm not going to talk about today, basically on thermal niches of yeast and what you can learn from studying them in their sort of native thermal environments. The first person I went to ask, like, which species should I use was Chris. He gave us some great advice. It turned out, we published that paper now a couple of years ago. The other thing I think that you should know is that it is true, everything he said about the faculty job market. And what I just want to say is that I had a top choice. That was MIT. They did not give me the offer. I had a second choice. That was UW-Madison, where there's a joint search in biochemistry. Betty Craig was chair at the time, and John Yin in systems biology. I didn't get the job. I was devastated. I had to settle for the University of Chicago. So there you go. So very, very pleased to be back here today. It's like such a great community. I have such like old friends. Actually, Anna and I had seen each other a zillion places, but never actually met. So this was a real joy to finally get to hear all the things that you're up to specifically, and then like the whole community. Students are great. This is awesome. Okay, now this whole thing the whole rethinking stress granules. I really will try to pay that off to you. I am as Chris sort of mentioned, a biochemist who you might have worked at has never actually taken a class in biochemistry. So all the real work and this detailed work is done by this like squadron of actual biochemists. These are a couple of the squads who have contributed to this work that I'm going to talk about today, which was started 14 years ago by a postdoc who is now tenured faculty at the University of Edinburgh, right? So this is a super, super long range project and was eventually pushed over the line by Hendrik, Jared, Caitlin, and Karen. So Hendrik is now back in medical school. Jared has his own faculty position at Texas A&M. Caitlin's in her faculty position at North Park University. And Karen happily still in the lab. She is an absolute killer. That's her right in the middle of our top picture. Okay, so right. The basic question I want you to think about is this. So we proudly study yeast. People are like, why did you choose it as a model organism? I'm like, well, I didn't. I chose it because it's an organism. It's great. It's like free of ethical constraints, right? But you have to sort of wonder, what is it trying to do? And what is it, what does trying to do anything mean in biology? It's like, from an evolutionary perspective, what are the sorts of things that are going to lead to more yeasts in the world? So we're very much in love with what yeast is actually doing and thinking about, right, in its single cell kind of way. One of the things that yeast think about a lot is how to adapt to their environment, right? And one of the most common things that you will see is there, by the way, a pointer of any sort? There may be. I don't think this is. Is there any sort of thing? My arms are long, but maybe not quite that long. Yeah. Don't know. Okay. So I'll gesticulate and use words, which I'm highly paid to do. All right. So on the left here, you have a budding yeast cell with a bunch of molecules floating around in it, RNA, protein. Then some sort of stress hits the cell, okay? Some environmental change that is sudden, probably maladaptive, okay? So we'll just call those stresses. We can have some long discussion about what that word exactly is supposed to mean. You see that a bunch of stuff clumps up in the cell, okay? And then there's some stress response and new genes are turned on and protein molecules that are these, oh, perfect. Thank you very much. Protein molecules that are molecular chaperones that help proteins to fold and so on, right? Like nobody knows exactly what they do or how precisely they work. They certainly are synthesized in massive numbers after many stresses. They get upregulated, and then in the fullness of time, homeostasis is restored. Okay, this is observed in all eukaryotic life. It's actually observed in much bacterial life. And I don't know, do you have any idea of like what's going on here? So I'll tell you that out in the world that I've grown up in, this is basically what we have been thinking. That this thing is a stress, some maladaptive environmental change. This is protein aggregation, which is bad, and it's toxic to cells. And so cells have to do something to save themselves from death. So they make a bunch of chaperones to try to clean up this mess, and chaperones are able to do that. Okay? Now, over the past 10 years or so, we have written a bunch of papers, done a bunch of studies, not in that order, that offer a completely different way of looking at this. Okay? And I just want to like get you in the right frame of mind right now, which is this is one of many possible ways to look at what is happening. I'm going to tell you all the phenomena actually do happen, but the interpretation can be completely different in a really fundamental way. Okay? And that what we've uncovered is, in fact, in many cases, this is just some signal, not a stress, right? It is something that cells know how to respond to, right? And have learned how to respond to, learned in an evolutionary sense, over a couple billion years. And they know exactly what they're doing, not surprised by this at all, right? For temperature fluctuations in particular, like, are you really telling me that they have no idea what to do and have to like unleash a whole stress response when you raise the temperature a little bit? Give me a break. Okay. And that this is not aggregation in the sense of it producing a bunch of horrible toxic stuff. It is what we now understand to be a process called biomolecular condensation. Okay, now that's a very big catch-all term. So I'm curious, how many people have heard of the term biomolecular condensation? Okay, maybe half of people. How many people have heard of the term phase? Superior laser. Yes. That won't be annoying enough. Thank you very much. Um, okay. How many people have heard of the term phase separation? Okay. Oh, even more people. Great. So here's one thing just to know. Back in the day, when Cliff Brangwyn and Tony Hyman were pressing on P granules in C. elegans and so on, right? They discovered this process where a bunch of protein molecules could come together, protein and RNA molecules would come together into some sort of droplet-like thing that had liquid-like properties. Okay. And there's a way to understand how something can go from a liquid phase into a liquid phase. It's called liquid-liquid phase separation. Okay. It's like a phase transition, only there's no transition. And a whole field sprang up around this. It's an incredible, like, new way to look at how cellular matter is organized in time and space. However, the more we worked on these things, the more we as a field started to realize that phase separation was just one of any number of ways that you can take a bunch of molecules and push them together, right? In a way that's going to change their biological activity, localization, and so on. Okay. We realized we needed a broader term to talk about this because phase separation is actually a very specific thermodynamic way in which to have this process happen. We started to realize that there's nothing special about the liquid state, that in fact, solids can be just as useful as liquids can, as those of you who study the cytoskeleton might know. And so we needed a catch-all term. That term is biomolecular condensation. Okay, so if you know phase separation, you're like, what is biomolecular condensation? You can sort of think of like, one is the umbrella term for the other. Hopefully that is useful. We're going to talk a lot about this sort of stuff. Okay? And that this is not cleanup. The chaperones, actually, the puzzling thing about chaperones is like, what do they do? How do they work? What are their substrates? That in many ways, we have been able to demonstrate that this is the answer. They are the regulators of this process. This is their day job. They don't wait around for horrible emergencies to break out in the cell. They're constantly remodeling condensates. So we have a paper from a couple of years ago where we show that, aggregates that, physiological aggregates that actually show up in the cell in response to heat shock are dispersed. They are first phase separation plus gelation plus other biomolecular condensation processes when they actually occur. And second, that the molecular chaperones can disperse these things 2 orders of magnitude faster than they engage with toxic misfolded aggregates. Okay, so this is actually the thing that the molecular chaperones appear to do most of the time. Like I said, no new facts, just new interpretation. So one of the questions that one might have, that certainly we've had, is like, well, what else is going on here, right? What is all this stuff for? Why is the cell doing this, right? Nothing about this tells you why the cell would want to do this, just provides another way of viewing what that phenomena is. Okay, so if you're here, you've probably heard of stress granules because you walked into a seminar called Rethinking Stress Granules. And if that's all that you've heard, you're okay. Okay, stress granules are these clusters of messenger RNA and protein that occur after stress in all eukaryotic life. Okay, and they look kind of like this in various situations. I'm showing you temperature stuff, but we'll talk about some other things as well. So you have some, in human cells under normal conditions, you have proteins and messenger RNA that are largely dispersed. Poly-A binding protein is one of our favorite proteins that binds to messenger RNAs and is also a core marker of these stress granules. And if you heat up the cells, then you see the clustering of these proteins and this co-localization with messenger RNA. Okay, these little dots are stress granules. The same thing happens in fly cells, right? S2R cells exposed to both heat shock and oxidative stress. So arsenic is a very common thing people dump on cells to cause them to form these kind of clumps. The same thing happens in yeast cells where you see them. It's a little bit hard to see because this is such a bumpy image, but it's from Bernd Pukow's lab, poly-A RNA and poly-A binding protein, which will co-localize into these puncta after a 46-degree stress. For yeast, 46 degrees is hot, but not too hot. They can totally survive this. They don't like it. This is a 10-minute treatment. A lot of these things will happen very, very quickly. Okay, so that's what we're looking at. Stress granules, clumps of protein and messenger RNA that you can see, that you can resolve by a microscopy. And that practically is like all we know about them. So if you ask me, Alan, what do they do? Be like, we don't know. Do they have a biochemical activity that's associated with them? Well, no, we're not one that we've been able to find. Well, what have you found when you've purified them? I'm like, well, some people have purified them, but given that there's no activity, right? There's no phenotype for knocking them out, right? One of the questions to ask anybody who says that I've purified stress granules is, well, how do you know? Right? It's like my dog has eyebrows. How do I know when I've shaved them all off? Right? You have to have some other way to tell. Okay. That is a funny thing, right? As a biologist, to be spending your life thinking about a physical phenomenon that's only defined by a technical limitation, which is the diffraction limit of a microscope. Okay. So we're going to make a lot of hay out of that today, like just to give away all the punchlines. There's tons of stuff that's happening sub-diffraction here. In fact, we think almost all the interesting things, and certainly all the things that people thought can be attributed to these large blobs, can be actually attributed to stuff that's happening pre-stress below the diffraction limit. Okay. And that these things are absolutely everywhere. Okay. So great. There was an early result, a bunch of early results, like 20 years ago. So stress granules were discovered depending on, which time you want to directly identify as like what we think of now as stress granules. There either was an early discovery in the 80s in plant cells, which then turned out to sort of not be quite exactly the stress granules that we talk about today. They were really brought into our current understanding of what a stress granule is about 25, 26 years ago. So, and that's by Paul Anderson and Nancy Kadersha. Okay. And so they even at that time had this amazing result that you might think is pretty sensible, which is that some transcripts, some messenger RNAs are induced during stress. Everybody knows this, right? There's a transcriptional response that follows stress, right? So the cell says, I was doing something before, I need to do something else now. I turn on a bunch of genes, some of them massively, right? Like heat shock will stimulate 1,000-fold production of new transcripts. And that these transcripts, unlike most messenger RNAs, right? I showed you the poly A messenger RNA was co-localizing with poly A binding protein in these granules, that these new messenger RNAs were excluded from stress granules. Okay? And that, if you think about it for a little bit, you're like, well, that could be very important. Okay. So that was also shown, that's for HSP70, which is a heat shock protein here. You can barely see this, but that actually says HSP70 messenger RNA. And I want you ultimately to like squint at this as long as you want, then I'll tell you something about this figure. This is showing essentially the same thing for HSP90. You can squint at this all you want. What I'll tell you is that these results are there. This result has been cited hundreds of times. There is no methods section in this paper. This is the only result. Okay. The more you squint at it, the more you're like, I'm not, maybe that's what that says. But it was clear, like people were seeing stuff. Okay. And they were reporting it. But this was before any sort of high throughput era arrived. Great. So then the high throughput era arrived. And people were then able to go, all right, let's isolate stress granules. Now, here I want you to have in mind my dog. Okay, what it means to actually isolate stress granules. And then doing things like sequencing the messenger RNA that you see, right, in those granules. Because now we have sequencing. And what they found was not that like, pre-stress messenger RNAs go to stress granules and new things are excluded, but instead one very, very simple message, which is that long messenger RNAs go to stress granules. These are data from one of the key papers. This is from yeast. This is also shown in mammalian cells. This has been seen in a bunch of other circumstances. So this is the longer the messenger RNA is, every dot here is a transcript. Here is the stress granule depletion score. So the higher you go up, the more things are not found in stress granules. The lower you go, the more things are found in stress granules. You can see, first of all, there is no sharp division in these categories, right? It's kind of a big cloud. And the reason there are these divisions is because that is one standard deviation away from the mean. It's not that there's some like natural cluster into which these things fall. But also, you don't need statistics to tell you that there's a negative correlation here, right? And if you look at this population, it tends to be longer than this population. Not hard to see. And that if you then do something in mammalian cells and put a stress granule protein that's known to phase separate, right, in vitro, and you incubate it with messenger RNA that's very long, then it will form these nice little droplets. And then if you incubate it with messenger RNA that's shorter and shorter and shorter, eventually those droplets go away. Okay, and maybe these things are sort of like stress granules, even though we have no idea because there's no activity assay and there's no anything else. It's like just one protein, bunch of RNA. But it kind of looks like, these things are droplets and aren't stress granules droplets. So maybe they're the same thing. So we should just basically be like, look, long messenger RNAs go to stress granules, just like we saw in previous studies. Okay, great. So that happened. And this is so clear and so sort of biochemically obvious that this should happen because, I mean, messenger RNAs have all this propensity to interact with proteins and other messenger RNAs, right? So the longer you make them, then the more probable, if there's a fixed probability per site, right? The more probable that it's going to participate in one of these large clustering interactions. That's called multivalency, for those of you who have heard the term, right? It makes a lot of biophysical sense that this would be the case. And so this sort of confirmed everybody's intuition that this is exactly what should be happening. What's more, there's like sort of biological intuition as to why this should happen, not just a biophysical intuition, which is that why does this happen after stress? And that's because when stress occurs, one of the things that happens in all eukaryotic cells is you get cessation of translation initiation. Okay. And then ribosomes will run off. So you get free messenger RNA. And then that free messenger RNA exposes all of these sites that are now have this propensity to interact with other proteins and RNA. Okay. And so the long ones preferentially, but everything to some degree, maybe that's why that you get that sort of smooth distribution, will undergo both RNA-RNA interactions and protein-protein interactions. And we'll go to these giant things where if you stand far enough back from this, it's just like some blob that you see in your microscope. Okay? And this sort of stuff was so influential that you can then go to reviews that say, what do we know about RNA? What do we know about stress granules? Well, in stress granules, what you have are long RNAs. And that's it. Now, of course, I'm telling you all this because it's totally wrong, right? And I'll try to tell you exactly why. But here's sort of a picture, right? Some small percentage of messenger RNAs go to stress granules. It's length dependent. There's no stress specificity. It's mostly biophysics and aggregation with some, initial process that leads to exposure of that RNA. And just to say that, like, this is not what was seen before. Let's go now take these data and overlay the 15 or 20 most upregulated genes in response to azide stress, which is this stress. So here they are. And I hope you can appreciate that, like, they're just a splat right in the middle of the distribution, right? There's absolutely no sense in which they're systematically depleted or systematically enriched or anything. I mean, they're all supposed to be up here if you believe those early studies. This basically said those early studies must be wrong, right? Nice idea. It's not what we see. Okay. Great. So we've been interested in stress-dependent condensation for the longest time. And we tend to look at these things a different way than a lot of groups. One of them just has to do with the fact that we're terrible microscopists. And also I'm employed by a biochemistry department. And because I have no training in biochemistry, I had to sort of show that I was a true biochemist, right? It's just willing to grind up whatever organism came my way and put it on a column, right? Spin it down, so on, right? Isolate whatever components. And so from the earliest days of my group, the approach was basically like, how can we, it's really hard to do microscopy to discover the components of stress granules. You'd really like to have something that was either sequencing based or mass spec based if you're right, RNA or protein. And so how do you actually do that? right? How do you do mass spec on something that's only defined microscopically? Okay, well, one of the things that you do is just go, well, I don't care if it's microscopically visible. I just care if there's a condensation process happening, right? Or something is going from a small particle to a big particle, and I'm not going to be particularly precious about exactly what those big particles are. All right. So here, by imaging, you can see exactly what you've seen before. You have a bunch of stuff in the cytosol of yeast, that by this way, these big blobs in the middle of the cell, the nucleus and the vacuole. And then in the cytosol, right, this lit up part, you get these little clusters. Okay, that's a formation of some sort of particle. So now imagine that if you grew up cells and then you lyse them and then you spin down a bunch of debris and then you isolate the supernatant and the pellet. And you do this twice. I mean, do it more than twice if you're doing replicates like you should. Good boys and girls. Um, you do it pre-stress and then you do it post-stress. And you ask, can we find proteins or RNAs that are in the supernatant before stress and then form large particles and go to the pellet after stress? Does that make sense? Yeah, okay. And what's nice about that is that you can take this whole pellet and this supernatant and you can put them on a mass spectrometer and ask what things are enriched, right, in each one of those things. You get thousands of proteins in a single experiment as opposed to having to go do microscopy. Then you go back, you tag the things and go do the microscopy. We've done that. This is actually a 2015 paper when we initially did this. And here you can sort of see by Western blot what this is supposed to look like. Okay, so here's at 30 degrees Celsius, a total fraction being probed for poly A binding protein, which is PAB1, anti-PAB1 antibody. Here's the result, the pellet after an 8,000 G spin, 100,000 G spin, and a supernatant. Okay, so this 8,000 G spin is going to get you really big stuff. The 100,000 G spin that's going to get you the slightly smaller stuff, but still bigger than the soluble material. And the supernatant is whatever remains, right? Hopefully that's mostly the monomeric species. So at 30 degrees Celsius, poly A binding protein is mostly in the supernatant that we expect. And then after a 46-degree 8-minute heat shock, it's almost entirely in the pellet, even in like pretty large stuff that you can sediment at 8,000 G. So now just imagine having those data proteome wide, right? We got a whole proteome for these things. And I'm going to spare you a bunch of stuff because I want to get to the RNA side of these things. What we discovered is that there's actually a lot of this stuff happening long before you can see a stress granule. And what that led to, that and a bunch of other results led to, this idea that condensation actually precedes stress granule assembly. And that there's a bunch of things going on, not just one thing. So imagine that you have proteins engaging in interactions with RNA in various ways, and RNA actually interacting with itself. These things can do all sorts of interesting condensation activities that are all subdiffraction, right? You'll never see them as some giant focus in the microscope. And then these things actually, by active cellular processes, like cytoskeletal transport and things like this, get drawn together and tethered to membranes and organelles, things like this, right? And that is actually what we see as the stress granule. And why do we think this? It's because mammalian cells 20 years ago, people were knocking out, not knocking out, they're poisoning microtubules and realizing that led to fragmented stress granules and other things that like indicate that there's some active processes that are bringing these things together. And only this stuff is visible by standard microscopy. This stuff, you just see a haze. You would conclude that nothing had occurred. Right? You would conclude that the dog had not been shaved. I'll stop mentioning my dog. All right. So what do we do now? Like, let's go look at this, but look at it on the RNA side. Right? Let's ask what happens to the messenger RNAs. So this is stuff that was done by these microscopy that's done by Caitlin in the group, just to show you that here's 30 degrees Celsius yeast cells, poly A binding protein here, poly A RNA here, and the merge of the two here. They're just both a haze at 30 degrees Celsius, as you expect. At 42 degrees Celsius, which Chris will tell you is not a bad heat shock, right? I mean, most people are studying heat shock are studying at like 37 degrees Celsius, maybe 39 degrees Celsius, but 42, heavens no, right? Like far too severe. No stress granules. And everybody reports no stress granules. You push them to 46 degrees Celsius, as I've shown you before, then you get nice little stress granules that are co-localized, messenger RNA, protein, blah, blah. Okay, next one. Do the same thing, except rather than doing mass spec, you do qPCR or you do RNA-seq, right? Supernatant pellet. And here is what you get at a very high level. So I'll show you the protein results that I've been talking about before on the left and the RNA results on the right. Here are the three temperatures. Okay, 30, 42, 46, 30, 42, 46. It's nice to be an old person in science because you get to like accrete results that all interdigitate with one another over time in a way that's like very difficult to do within the space of like a 4 or 5 year PhD, right? You guys are what, fourth, fifth year? How many graduate students here? How many people passed your third year? Oh my goodness. Two, really? Just three of you? Well done. Yes, like, what are you doing with the rest of them? It's just, you guys still have the joy of science. Am I right? Still have seminar? You're supposed to be, aren't you? Yeah, okay. Right. So these things we collected, 15 years ago, 10 years ago, okay, protein data. This is the mass spec data. This is proportion in the supernatant. Okay, so all the way up here are soluble proteins. And if you see them remaining soluble all the way across, then nothing is happening to them. Here are things that are in the pellet, right? So membrane proteins will just always be down here, okay? And nothing is happening to the, in fact, if you look at the distributions here, these little funny, double lozenge shaped, quasi-violin plots, it doesn't look like anything is happening. And that's because it's just a small proportion of proteins that are doing this condensation process, right? So anybody who tells you there's like massive protein aggregation happening when you treat cells with these sorts of temperatures, false, right? Empirically false. Instead, there's this tiny, tiny subset. And poly-A binding protein is one of these things. So if you track poly-A binding protein, boom, it goes like straight to the pellet, right? Because it specifically condenses. And by the way, it does this in vitro alone with no other cellular machinery, right? It autonomously senses temperature, undergoes phase separation condensation process, right? Doesn't take RNA with it. RNA actually inhibits that process. So it's not responsible for any of these things either. All sorts of fun stuff. All right, so that's the protein case. And you can probably see that is not the messenger RNA case. Messenger RNA case, totally different. Same temperatures, same organism. But here, most messenger RNAs are soluble with a long tail, which we'll talk about, before stress. And then you've taken to 42 degrees Celsius, boom, like almost everything starts moving down, right? Getting a higher proportion in the pellet. And by 46, almost the entire thing is down towards the bottom of the tube. Okay. And the proper way to read proportion in the supernatant is not that they're sort of in an intermediate point in the tube. It's just like the fraction of things that are absolutely at the bottom of the tube is being weighed against a smaller fraction that remains floating at some indeterminate point in the tube. Okay, so it's really a two population kind of measurement. All right, so this is totally different. And by the way, totally different than what we were expecting, because you remember this previous result that said that long messenger RNAs go to stress granules and short messenger RNAs don't. Well, this is already completely incompatible with that result, right? Okay, so what on earth is going on? And the other thing that I think is really important is that what's going on here? These are stress cells with a bunch of RNA and protein condensation, but there are no stress granules. So what are we seeing here, right? How are we supposed to talk about this? The only definition of a stress granule is something that appears as a focus in my microscope. So the point is, this is subdiffraction stuff that's just happening, and it's the same proteins, right? Poly-A messenger RNA and poly-A binding protein and a host of other proteins. They're doing all the stuff, but you can't see it in a microscope. I mean, you can't see it in a typical fluorescence microscope, right? In which 99% of these studies are done, right? And screens are done where if you make a mutation genetically, right, that's supposed to knock out stress granules and you see that everything is a haze, you conclude that stress granules have been knocked out, right? But you can now see that you would be utterly wrong to say that the condensation-related stress response involving mRNAs and proteins had been knocked out, because that is happening whether or not there's a haze. In fact, this is not just a heat shock case. then did azide treatment, which is oxidative stress, ethanol treatment, which yeast hate at high concentrations, even though they make it. And there's always a concentration that we can find, which people would consider to be a pretty significant stress, okay? Where like you're not allowed to use this in the lab without protection, right? And yeast feels the same way about it. And yet there are no stress granules in any of these conditions. You push the system a little bit more, and then you see stress granules in all of these conditions. If you quantify stress granules, sure enough, right? It's just like flat line until you reach the higher concentrations. But in every case, the lower concentration, if you will, of temperature, right, is inducing a significant amount of differential sedimentation, which is now going up on this axis, confusingly. Azides, low levels, higher levels, right, are all above zero here. Ethanol, same thing. Okay, so condensation without stress granule formation across a range of relevant stresses. So what's going on with length? Well, here's the same data I showed you before, those three strips, only now I've blown them out on the x-axis, which is length. Hopefully you can appreciate that, first of all, there's things that you've seen before, which is the entire transcriptome moves down in response to stress, increasing stress, right? Do you remember that long tail on the unstressed messages that we saw before? This thing, right? Why is that so long? Well, now you can see why it's long. It's because there's this really funny thing, like there's length effects all over this graph, right? Here's mRNA length. But one of those length things is in unstressed cells. And it's that long messenger RNAs have a lower proportion in the supernatant. Are they condensed pre-stress? No, they are long, and long messenger RNAs are huge. And if you put them in a centrifuge, they will sediment like any other large molecule, right? So this is actually what you expect, and I'll convince you of it. I hope I will convince you of this, right? Like, so this is completely normal behaviors for some large biological molecule. Messenger RNAs or mRNPs, right? Like the messenger RNAs decorated with all the proteins in the cell are just like so much larger than any protein that all your intuitions about what proteins should be doing in a sedimentation experiment just go right out the window, right? You're now encountering like the Death Star, right? Every time you encounter a single messenger RNA, that's no moon, right? That's an mRNA, or mRNP, sorry. Okay, so this thing is clearly going to be an issue. And then you can see that this same sort of pattern is just ported down, right, as you increase stress. Okay, well, this is why the previous study went off the rails. Would you believe they didn't do a non-stress control? So you'd never see this. You wouldn't see that there's no stress-dependent, length-dependent effect. You just see this and conclude, right, that long things go into stress granules. Do your controls, people, right? The biology utterly changes as soon as you see this. Okay? Not only was the previous conclusion just not supportable the minute you do a non-stress control, but as I'll show you, you see something that's much cooler. Right, just to say, if you do the simplest possible thing of like trying to fit what a, two-parameter model of mass coupled with viscosity of the fluid and blah, 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 you actually just do sedimentation theory to try to figure out what these curves should look like. Like that's what it should look like. So pretty good. And if you then say, okay, well, let's just do a simple model of condensation. We think it's multivalent, right? We think there's supposed to be interactions that are happening along the length of the messenger RNA, right? Sequence-mediated interactions. So just imagine that you have some like equal probability that per unit length that some interactions are happening in messenger RNAs. But now add in something that's been missing, I think, from the intuition for a lot of these studies, which is that there's another way to interact. I mean, I just showed you essentially that this condensation is length independent. How on earth do you get length independent condensation when all of the interaction propensity comes from long messenger RNAs? And of course, the answer is that messenger RNAs, regardless of how long they are, always have two ends. And so if you have end-mediated interactions or end-mediated condensation, it will naturally be length independent. Okay? So add in another way to have interactions. Here I'm showing it at the 5' end because mostly that's where we think the action is, but this is in principle also just the other end has no effect on the models. Now just fit the models, which I'll flash some math, but it's just like, oh my gosh, it's Poisson. Oh heavens, right? Like, can I even write down a Poisson model? It's like not hard. And then realize that like, here's what you get from just simple sedimentation theory. Here's what you get from a model that is like a term that is length independent and then a term that is length dependent. Fit these, like it's a 2-parameter plus 2-parameter model for the entire transcriptome, right? So you're just trying to capture the average behavior, pretty good. Not so bad, right? That's basically what we see. And what we see is that that first component, right, which is the length independent, sorry, the second component, the length independent component is by far the dominant contributor to this condensation. And you don't need a model to see that. The way to see that is just to look at the shortest messenger RNAs. Okay? So if you have very short messenger RNAs, then the length dependent condensation is going to get weaker and weaker and weaker. And eventually, in the limit of 0-length messenger RNAs, it will vanish entirely. And so you should get some sort of plateau that tells you what the isolated contribution of the length-independent end-mediated term is. Now, I hope you can see that as you follow these things back, where this has no condensation at all, it's just like the behavior of free messenger RNA in the centrifuge. Okay, follow this back, and whether you follow the model or you follow the average behavior of the transcriptome in gray here, these lines, by the way, that's the marking the bottom 1% of data. This is the top 1% of data. So between these lines, it's 98% of the data. Okay, it's like basically everything. What you can see is that there is a plateau, and that this plateau is at different places depending on the severity of the stress. Now, if you think, what should happen if this is all being mediated by length-dependent effects, then all these things should go back to 1.0. And they should fan out, right, from 1.0. And that is not at all what you see, right? For the shortest possible messenger RNAs that we observe, and what appears to be like the limit of 0-length RNAs, you still get a massive component, and that's exactly what you expect, what you need to have from end-mediated condensation. Okay, so this is telling you that you get most of this big drop just from end-mediated effects in this model. So a demonstration that directly has to be only this way. I haven't shown you some proof that this is like the unique model that can fit this, but it is super simple. Okay. And as you can probably guess, there are really simple reasons to expect that this is a very good model for cells to have, because then we can do the next thing. We should just ask, well, if I compare the tendency of messenger RNAs to sediment to the amount that they're being induced in response to heat shock, what do you see? And what you see, I'll give you yet another confusing graph to look at, but I'll immediately hopefully explain that after taking into account all these length effects, which you have to do, right? You can now quantify to what degree do individual messenger RNAs escape from sedimentation, okay, relative to their 30 degrees Celsius numbers. Okay, so that takes the 30 degrees curve and like does all the differentials properly, right? So you can get a length independent number here. Okay, that's here. More escape, less escape, right? So more condensation down here, less condensation up here. And here is just the full change in the messenger RNA abundance at 42 degrees Celsius versus 46. Now, I hope you can appreciate that there's just a lot going on in this upper right-hand quadrant of induced escaping messages. And for those of you who work on heat shock, you see a bunch of your like happy usual suspects. Okay, SSA4 is the stress-induced HSP70. HSP104, the big thing that actually pulls these condensates apart, right? If it's put into a test tube with HSP 40 and SSA 4. HSP 26, little small heat shock protein, massively induced in response to a wide range of conditions. This is true, only less so at 46 degrees Celsius, where the cells in our hands just seem to start battening down hatches. It's a very severe stress for them, but not lethal. Okay, well, this is what was reported 25 years ago in those low throughput studies. If you have a length artifact interfering with all of this, you don't see it, but the minute you control for it, it immediately appears. What cells are doing is changing their mind. They take all of the messages that they were working on right before stress, they mash them away into, I'm not going to show you today, but it's in the paper that we just published, non-translating assemblies, okay? Except they then preferentially exclude the new messages that they're expressing in response to stress-specific messages, by the way, and selectively translate those so they don't have to dilute those messages into a giant bath of irrelevant crap that they've been working on minutes before. Their game is really, really simple, right? And then hold on to the messenger RNAs so that as soon as you're done with the stress response, you can get all of them back without resynthesizing your entire transcriptome. Okay, very, very smart, unlike the other model in which cells are stupid, right? And like overwhelmed with toxic crap, right? The minute the tiniest perturbation to their temperature occurs. Okay, so great. That's, I think, fun biology that is like there for the taking. So now what actually causes this? I'll tell you, we have no idea, right? Something has to be marking these messages, presumably in the nucleus. That would be the way to do it, right? If you were a smart cell. We don't know what that nuclear mark looks like. I can tell you we have, of course, tons of hypotheses because, like, you know, who doesn't? I'm paid to do that. But we have no idea whether any of them are right. All right, so now why do you think things condense? And why do they do it across all these stresses? Well, I told you about that model where, you know, ribosome-free messenger RNA was the thing that caused stress granule formation, right? You expose, you know, ribosomes run off and you get all that exposed messenger RNA. And then that messenger RNA is a template for all these interactions that if you stand far enough back from them, look like stress granules. Okay? This. And sure enough, if you know, look at, I'm not going to train everyone on polysome gradients, but for those who've seen a polysome gradient before, and you ask what happens during heat shock, okay? These are ribosomal subunits, and here are like two ribosomes, three ribosomes, four ribosomes bound to messenger RNA. Okay, under normal conditions, they'll pile up into these giant things, but if you heat shock cells, right, continuously down to 46 degrees Celsius, this is data from Bernd Bukow's lab, by the way, not our data, you'll just flatline those polysomes. Like all the ribosomes run off messenger RNAs, they all accumulate as ADS-free ribosomes. Okay, so we know that in fact this does happen, right? Ribosome runoff actually does happen. Cessation of translation initiation really does happen. And another reason that people had concluded that it must be that ribosomes are really like ribosome-free messenger RNA is the substrate that causes stress granules, is that if you treat cells with a drug called cyclohexamide that locks ribosomes onto messenger RNAs, despite the fact that they're no longer translating, that stress granules disappear. Okay, so this is 46 degrees Celsius, a very strong heat shock that will cause stress granule formation. As you can see, no cyclohexamide, you add cyclohexamide and stress granules disappear. Okay, so this is, again, additional evidence that if you kept the ribosomes on these messenger RNAs, they wouldn't be able to form stress granules. All right, so the group did this, built this crazy panel of reagents that I just think is really great. They looked at translation initiation. They identified a bunch of factors that they could tag with a degron, okay, so they could degrade each one of these translation initiation factors in turn in separate strains with auxin. And then just asked, well, see, that should break translation initiation at a bunch of different steps. Okay, and maybe there's some special step where we can find that like condensation either does or not does not occur. It turns out EIF3B, which is like the B subunit of a 5 subunit complex in yeast, the EIF3 complex. If you deplete EIF3B, here's the transcriptome P soup. Now, whatever its odds, it doesn't really matter, right? Some giant cloud. You can see the long things are sedimenting more than the short things, as we expect. Now, if you deplete EIF3B, you get the gold distribution, right? The whole transcriptome shuts down. And this is no heat shock, no, it's stressful, of course, right? But you're just blocking translation initiation specifically by depleting that factor, and you get transcriptome-wide condensation, okay? Totally as we expect. If you do this with a bunch of different factors, then the more you're actually able to deplete those factors, you get decreases in condensation. There's no factor that we can identify that's particularly special. Basically, blocking translation initiation at any step causes this condensation to happen. What's cool, though, is that that does not cause stress granule formation. Okay? So here's mock treated cells that you've seen before, 30 to 42, 44, 46, they start to form stress granules. Here's EI3 to B depleted cells. No stress granules, no stress granules. You can now see stress granules. Now you can really see stress granules. Stress granules, and you just don't see them. The median still at 44 is still at 0. You quantify these things, but tons of condensation. Okay, so the change in sedimentation is going up. If you inhibit, just like I showed you, right, the whole transcriptome moving down, that's now this distribution moving up. Increased sedimentation, same at 42, minus plus, right? It causes condensation no matter what the background stress is. So yeah, I hope you're having this feeling now. Which is like everything I told you is actually false. It's just flatly false, right? It's not true that if you have ribosome runoff, right, an exposure of messenger RNAs, that you get stress granules. That is not true. It's empirically not true. It's just not true that long RNAs go to stress granules. It's just not true, right? Like, I don't know what to tell you. I don't know how we got to this state, but I have to be like the one to break it to you. Like, doesn't work that way. What is the alternative? There's a bunch of stuff happening though, right? So what do we call these things? I don't know. We had to come up with something. And so I just figured like, they're kind of small, right? They're subdiffraction. They're everywhere. Let's call them ticks. Translation initiation inhibited condensates. Okay, they're what happens if you block translation initiation. They're length independent. They happen in multiple stresses. They're subdiffraction, right? Basically, what you're looking at is something that looks like this. You have messenger RNA. If it becomes, it can be participating in translation. If it's blocked in initiation for too long, something we have no idea, but probably a protein factor, because it's hard to imagine that the caps of the RNAs are directly interacting with one another. This is the kind of thing that proteins are very good at doing. Right, grabs this thing and pushes it into a subdiffraction condensate that may have a small amount of RNA-RNA interactions, but it's mostly protein mediated. Okay, small and just gets collected here, and it can be reversed by molecular chaperones. All right, and that these things are actually happening all the time. If you go read our paper, these are present in unstressed cells. Anytime that translation initiation is transiently pausing, that message will be collected into these little things that you can sediment out, even if there's no stress at all and no treatment of any kind happening to these cells. Great. One last thing, just to convince you that there's really something going on here. I told you cyclohexamide blocks stress granules, right, by locking ribosomes on messenger RNAs. That you can see. Okay, so 46 degrees Celsius, you add cyclohexamide, no stress granules. What happens to condensation? It still happens. So 42 degrees Celsius minus cyclohexamide, huge, 5 standard deviation effect. Oh, we're now down to 4.5 standard deviations, right? There's some reduction, but there's absolutely no sense in which you're blocking condensation of messenger RNAs. And why does this make sense? Well, it's because ribosomes have nothing to do with the 5' end specifically. They can be in the body of the messenger RNA all you want, and they're not going to interfere with a protein that's specifically targeting the 5' or the 3' end of the message. Okay. And this has actually been shown by others, like many folks have shown that ribosomes block stress granules through a range of different approaches, actually quite beautiful approaches. Stephanie Moon has done this. a single Russian guy whose name I can't directly recall, but basically used all possible, in a sort of classically Russian way, all possible translation initiation inhibitors and showed that the presence of a single ribosome upstream on a messenger RNA was sufficient to exclude the entire messenger RNA from stress granules. Just beautiful work. All right, I'm going to skip over for time, showing you that we can even cause this to happen with specific messenger RNAs in unstressed cells, and just tell you we can cause this specifically to happen in unstressed messenger RNAs, in unstressed cells rather, with single messenger RNAs. But what do we think is going on? Okay, the thing I showed you before, formation of ticks, now in stress conditions just gets elaborated, because instead of having a small amount of stuff that's going in and out of the translationally blocked state, now you have a huge flood of stuff that's coming out in translationally blocked state. So you're going to get huge amounts of ticks, right? And almost all your pre-existing messenger RNA is going to go to this state because they're all going to be substrates, because the entire, right, as you saw from those polysome profiles, most translation initiation is shut off. But you still have enough that if you have now a bunch of extra free ribosomes around that have been freed, right, their attention is now not being drawn away by all these other pre-existing messages. If you're able to mark new messenger RNAs in some way that protects their cap from this condensing factor, then they're going to be selectively translated. We actually think we know what that factor is, at least one of them, which is, well, the nuclear cap binding protein just stays on under these conditions and can support translation, as everybody knows it can, because it is known to be able to do the pioneer round of translation. Okay, this is reinterpretation, exactly what is happening with putting that cap binding protein on in the first place. And then these ticks are just the precursors to stress granules. If you push the system hard enough, then the cell, for reasons that we do not understand, will grab all these things and put them together into foci. But now this really reduces the role of stress granules, right? Like now you have to kind of say, okay, what's the function of just doing that? Because there's all this other subtraction stuff that doesn't have anything to do with stress granules. So I'll stop with my little sculpture, guys, and thank you. Yeah.
Hi.
Hi.
I have a question about the future of the field.
I don't know if I can answer that.
Yeah, well, but it's maybe, I understand that you're not working with functions,
Mhm.
biological functions essentially, but maybe talking to other people when reading.
Yeah.
Because for many decades, people thought about free radicals as a bad thing.
Mhm.
And we know now that cells intentionally produce free radicals because they have biological functions.
Yeah.
It doesn't matter, there's no stress involved.
Right.
My question is, people in stress granules field
Yeah.
have been thinking about that you can have stress granules completely independent of stress signals, what we canonically think
Mhm.
as a bad thing for the cell.
Yeah. Yeah.
Right? Or it's just a response, for example, when temperature involved, it's just a physical, biophysical response of the molecules.
Yeah. Right.
Yeah, and I understand that the condensation thing is new, right?
I mean, it's not new to us. I mean, it's relatively new to humanity, right? But like,
So is there, so the question about the function of the
Yeah.
condensation
Yeah.
is also discussed. Does anyone have a clue about it?
yes, right. Great question. So in the broad field of biomolecular
Yeah.
condensation, what we see, okay, there's these sorts of RNA condensates. So we did just discover these things. And what's more, like, let's be careful, until we see these things, the vast majority of biologists are just gonna be like, well, but couldn't they be something else, right? Like, might there be some other thing? Like, I agree. Or like, we have to get single molecule visualization of these things to see exactly how big they are. I can tell you that we have published, even in the paper, a bunch of more things that will tell you like how big they are, right? They're as big as polysomes, but no bigger. So there's a reason you can't see them in the microscope. But what are these things doing? They're one of 100 different condensates that have been identified. This is the biggest question in the condensate field. What do they do? Right? So the classic thing that people have been thinking about is, well, it's obvious, right? They form a compartment, and inside that compartment, things happen. And that is one of like an uncountable number of different possible things that could be happening, right? That are the function of condensates. So another possibility is that by making a condensate, you now make it unable to transport across membranes. Or by making a condensate, you now create a surface that didn't exist before. Or by making a condensate, you are able to use a domesticated phase transition to sense the change in some very sensitively changing biophysical parameter, because condensates are just phase transitions which are the limit of infinite cooperativity, right? Even in finite system size. What I'm trying to express to you here is like the range of possible things that could be going on are the range of emergent phenomena. This is like thinking, well, I know about one water molecule, but so what does multiple water molecules do? And you're like, kiddo, sit down, right? Like we're gonna have to talk about flowing and splashing and dripping and wetting and all these other things that just simply don't exist. Freezing, right? Like there is no single molecule equivalent of this behavior we're about to talk about. And that's what's happening in the condensate field. What are even the range of possible functions we should be looking for? Totally unanswered question, makes it fun to come to work. I don't know if that really answers the question, but like big concerns and like lots of thinking about how we should even prosecute this. Yeah, Aki.
Very interesting. I have one question. So when you look at when we look at your figure, that the condensation and the temperature is very positively linearly correlated. So I'm just wondering how the temperature really determines the amount of condensation.
Yeah. So I think that you're talking about if I can here. Yeah, basically, is this?
Well, more later on that you showing that condensation is actually increasing without showing the granule.
Like
that's
this one.
It looks like
exactly. Right. So I mean, one way to think about this is that this is a steady state process that is being determined by two forces, the force in and the force out. Okay, the force in we control, and the force out is fixed. And so what you get, at least in some regime, is what looks like a linear response. Okay, what's the force out? We think that's actually just the native molecular chaperones that are sitting around that are just very happy to be disassembling these condensates as soon as they arise. But they, in stress conditions, simply get overwhelmed, right? That titrates them away from a lot of their normal activities, unleashes HSF1, does all sorts of other things, right? Like actually solves a ton of problems in the heat shock field that are very difficult to solve otherwise, because you need misfolded proteins that have never been found or other things. So hopefully does that get you at least a little bit of intuition about it?
Making the granule.
Yeah.
I think it looks like still that the lower temperature is less condensation.
Oh, that's true. That's absolutely true. Yes. So this is titratable. So maybe, another thing to say is that this is not a phase transition. Yes. Why is this? Right. So this is also a very fundamental issue in the condensation field, which is that I think there's intellectual damage that's done by rushing in and claiming that these things are, for example, phase transitions or phase separation, because thermodynamically, those are very specific claims, right? And it means that you're going to see switch-like behavior as soon as you cross a phase boundary, as soon as you exceed at critical concentration. But that's not how it has to work, right? Instead, this is all kinetically controlled, right? This is all through nucleation and growth. And so it's very easy now to have titratable control, which is what the cell wants, right? It's like, it wants a dose response curve, right, to these things and not threshold-like behavior. Yeah.
That's interesting. So that if you put the other stress, like, the same thing happening.
It's exactly like I showed you. In each one of these cases, they're all sub-diffraction, but like the lower level of ethanol, higher level of ethanol, right? Lower level of azide. No, not linear. I mean, linear is a very strong claim. So this just, if I show you 3 points, you're gonna be like, that looks roughly linear. But no, it's gonna plateau at some point. Yeah. It's just it's an S, of course. Like everything in biology is some sigmoid on some scale. Yeah. Here.
So I'm really interested in this end effect. And so I'm thinking about messenger RNAs that don't have an N7G cap.
Nice.
And
right.
Don't have a poly A tail.
Yep.
So they're like NAD can start some messenger RNAs and histone messenger RNAs aren't polyventilated. So is this something that maybe evolved to escape this stress.
Gosh, I'm completely in love with this idea, right? Like that this is a reason why you can have alternative caps or no caps at all. And like, there's a lot of things we would have to put together in order to say like, this is definitely what's happening there. But this is the place where my heart is at, for sure. Right. This is like, that's the way that you would want to regulate this is by tweaking the 5' end or the 3' end, right? Because that's the locus of control, for sure. Absolutely, for sure.
Hi, so I'm waiting for you to dismantle this possibility. So
Wait 20 years, and then I'll publish a paper.
in your discussion section of the paper you published about this work on the different stressors,
right?
You said that newly synthesized RNA and basically have an easier time of escaping.
Yeah.
The brand. And I'm curious, like, at what point is it just as simple as, oh, it's kind of like a rubber band ball, where the rubber band that's most outside the band ball is most easy to take off? Right?
Yes, right. So we went back and forth a ton about this in the group. Like, what is the simplest model that you can come up with that would explain this behavior? And I'll tell you that Hendrik, who's the first author in the paper, was very insistent there's an even simpler model than one that I've said, which is that when stress occurs, there is a limited pool of the condensation factor, whatever it happens to be. And it is absorbed as things condense. And the newly synthesized messages emerge after that has occurred because transcription takes time. And so they just emerge into a world in which the propensity to condense is much lower and they're therefore excluded, right? Now, I think that's a very pretty idea. To me, and the reason we didn't like plaster the paper with that is that it runs into empirical problems as you like step up, you do low amounts of stress and then you do higher amounts of stress and you start to realize that like there's still tons of capacity left after you do the initial stress, and yet still things are being excluded. And so it doesn't seem to be level of stress dependent. And so to me, it really makes it difficult to make those sorts of models work. That's not exactly the rubber band model. So we should probably talk more about exactly how these things, if they encounter a pre-existing condensate, are they less likely to join? But the one thing that I do to shape your intuition there is that if you're thinking about one condensate, then at some level you're already thinking about the old world in which small numbers of like countable numbers of condensates are around, versus these condensates are of order 5 messenger RNAs. So there's just tons of them all over the place, right? And presumably there's a size distribution of those. And so the idea that somehow they're going to bounce off everything at that scale, when it's like, it should be very easy to find a little chair in musical chairs, right? I don't know if it could be made to work. Maybe so. Great. Okay, I think we're over time, so I just at least want to allow people to run off to whatever it is they need to do. Is that true? Yes. Okay, so thank you guys very much.